Welcome, everybody, to this week's edition of the Extracellular Vesicle Club. This is an event that is endorsed by the International Society for Extracellular Vesicles. And today we have a topic that is close to many of your hearts. And it's going to be presented by a group that is among the leading experts on this topic in the world. And that topic is platelets. We've got tons of platelets in our blood. And sometimes those platelets are doing very good things, but occasionally we don't want them there anymore, especially when we're trying to study our extracellular vesicles. So we're going to hear today about some of the things that we can do to remove those platelets and some of the ways that we can analyze our samples after their removal. And so I'm going to hand this over now to Rienk Nieuwland. So Rienk is the head of this laboratory that has done so much exciting work on platelets and on EVs. And Rienk is also our chair of education at ICEV. So Rienk, thank you so much for joining today. Thank you and your lab members for presenting. And I'd like to hand this over to you now.
Thank you, Ken. So the talk of today will be presented by Jillian Bracht, who's a postdoc in our lab in Amsterdam. And the presentation comprises two papers. One recent paper that was published by Britta Bettin as a first author in the Journal of Thrombosis and Hemostasis, and a second paper where Jillian Bracht is first author that has just been accepted by the Journal of Extracellular Vesicles. And we thought that these two papers really fit nicely together, and Jill will present both papers today.
Yeah,
Jill, so I think the floor is yours.
Thanks for the introduction, and also thanks for the opportunity for us to present here today at the EV Club. So let's see, as Rienk mentioned, my name is Jillian Bracht, and I'm happy to present part of the research that was performed by the Vesicle Observation Center in Amsterdam. As Rienk also mentioned, I will present data of two papers, and Rienk and Britta are also here to join in for the discussion later on. So the first paper indeed has already been published last year, and in this paper we focus on the removal of platelets from blood plasma to improve the quality of extracellular vesicle research. And basically that first paper laid the grounds for the second paper. And in that one, we look at the platelet removal and what it actually means for downstream analysis. And in this case, we focused on microRNA analysis. So considering that blood circulates throughout the whole body and can be obtained in a minimally invasive way, blood-derived vesicles have become a huge topic, a huge interest. And in an ICEV rigor and standardization survey from 2019, it was actually found that plasma is currently the most used body fluid for EV isolation. And to prepare blood plasma, blood needs to be centrifuged. And the method of centrifugation knows many pre-analytical variables that can actually affect the composition of the obtained plasma. And by that, you can think of centrifugation settings, but also the person that is operating the centrifuge and therefore pipetting the sample, for example. So this is one possible centrifugation protocol that exists. It's also actually the protocol that we are using in the lab. So this protocol consists of two centrifugation steps of 2500 Gs for 15 minutes with no break on the centrifuge. And after the first centrifugation step, we take out the plasma from the top, we put it in a new tube. And what is important there is that we use these LIGO bricks to measure exactly 1 centimeter above the Buffy code so that we don't take additional cells with our plasma. Then we perform another centrifugation step, same speed, same time, again without the break. And again, we use this 1 centimeter and we only take the plasma above that line. So eventually, when the blood plasma has been centrifuged twice, of course, it contains the vesicles that we are interested in. But as many of you know, it also contains proteins and lipoproteins. But what we figured out is that actually this blood plasma also contains red blood cells. They call them erythrocyte ghosts, so they are empty erythrocytes, but also platelets. And today I will mainly focus on the remaining platelets that are still present in this plasma. So, as the title suggests, platelets. So just a brief summary of what they are. They are anucleated blood cells that derive from megakaryocytes. They are about 2 to 3 micrometers in diameter, and they have an important function in blood clotting. So here in the picture, you can see resting platelets or inactivated platelets, and then on the right are the activated platelets. And here I want to show you something that we call the protocol effect. So in this initial study, we used 2 different protocols, so different speed, different time of centrifugation, different break settings of the centrifuge, but also different with this distance from the pellet. So one is exactly 1 centimeter and the other one is more or less like by eye. And in this graph, you can see the concentration of platelets on the y-axis from protocol A and protocol B. And you see that the concentration of remaining platelets in that plasma can be quite different when we use a different protocol. And this table is an overview. I don't want you to focus too much on the exact numbers that are in it. But what I try to show here is I present 19 different protocols over the last 5 years. And these studies, they all focused on cancer-related EV microRNAs. So to develop biomarker assays, for example. And the important thing that I want to show with this paper is basically that there is insufficient reporting and reviewing. So everything that is here in red is either not reported or not performed. So there's insufficient reporting, but also reviewing, because of course, all these papers have gone through a review process. And there are 3 studies in which they don't even mention the whole blood processing at all. What I just showed you is that a different protocol means a different sample at the end. So there's different concentration of remaining platelets, different composition of the eventual plasma that was obtained. So there's no standardization at the moment of which protocol should be used for blood processing. And we believe that this could actually be one of the reasons that it's so difficult to validate biomarker assays at the moment. Since everyone is processing their samples in a different way, it's not weird that downstream we do not find the same results. And even with the same protocol, this could become complicated. So this is a figure from the first paper, so from the paper of Britta. On the x-axis, you can see the platelet concentration. Y-axis is the number of samples. And then 3 studies were performed. They all processed blood samples. And you can see that they find quite different concentrations of remaining platelets in those studies, while they use the exact same protocol on paper. The blue line here on the right, actually, it was figured out that the centrifuge break of the centrifuge was broken. So that is also something that they figured out because of this study. So there are, even with the same protocols, it's difficult to obtain the same sample, so to say. Here in black, the dotted line is actually the cutoff of a hematology analyzer. So normally when we measure the concentration of platelets in a routine lab, a hematology analyzer is used. And you can see that there are many samples that would actually be considered to be platelet-free by this type of analysis. In our case, we use calibrated flow cytometry to measure the concentration of platelets, which is why we can detect below that limit as well. So why is this important? Why do we care about this? Well, there's, of course, a consequence to having remaining platelets in your plasma. And the first one is that usually when plasma samples are prepared, they are often stored in the -80 in a biobank until further analysis. And what was figured out in that first paper is that once we freeze thaw our samples once, we are actually increasing the concentration of platelet-derived vesicles. So on the x-axis, you can see the platelet concentration of the samples. On the y-axis, the EV concentration. And then here in red are the platelet-derived vesicles, and in green, the erythrocyte-derived vesicles. And you can see that there's a correlation between the concentration of remaining platelets in our plasma and the eventually measured platelet-derived vesicle concentration. So it is likely that the platelets that are still present in our plasma are fragmented upon this freeze-thaw cycle. Another issue is a co-isolation. So if we have remaining platelets in our plasma and we use, for example, size exclusion chromatography, the basics of SEC are actually that particles that are larger than the pores of the resin that is being used, so in general, this is around 75 nanometers, they migrate faster over the column. Platelets, as I just told you, are around 2 to 3 micrometers in diameter. So they're actually migrating at the same time as our vesicles, meaning that we are co-isolating them at the same time. So this is true for SEC, but because of the similar density, for example, there's also a problem when we use other EV isolation methods like gradient centrifugation, for example. So yeah, we are actually co-isolating those particles at the same time. And as some of you maybe know, platelets can also contain microRNAs or mRNAs. So when we isolate them at the same time, they can also confound our downstream EV analysis. So a brief recap of what you really need to remember from this introduction. So the plasma that is centrifuged twice still contains remaining platelets. The concentration of remaining platelets actually depends on the protocol that we use, on the centrifugation protocol that we use. Those protocols are not standardized, and this can really affect either EV analysis, as I showed you previously, with an increase in the platelet-derived vesicles, but also the downstream analysis can be affected by this. So I already showed some of the data of the first paper during this introduction, but the goal of that study was really to investigate if platelets can be removed using filtration. So for filtration, we use 800 nanometer polycarbonate track-etched filters. I have some pictures here of the materials that we are using. So we use the filters. We have a filter holder and a syringe. The filter holder is opened. We place a membrane on top of it, add this rubber on top of it, and then we close it. We add the syringe on top, and we press the plasma through this system. It is important to use track-etched filters because they have a uniform pore size. So it's not an average, but it's the actual size of all the pores is similar, so that larger particles cannot pass. And what was found during the first study, this graph shows the concentration of platelets. And then here the left bar is an unfiltered plasma sample. And here on the right, well, it nearly disappeared, but you can see that we can really lower or almost remove all platelets from our samples. So very happy with that. But of course, we do not want to remove our EVs in the same filtration process. So here on the right, you can see the concentration of platelet-derived vesicles and erythrocyte-derived vesicles. And there you see that unfiltered or filtered samples contain the same concentration of vesicles. So this filtration step can remove the platelets without affecting our EVs. So a brief conclusion of this first paper. We saw that plasma still contains platelets after double centrifugation. Filtration reduces this platelet concentration without affecting the EV concentration. And therefore, in that paper, we also recommend to measure and report the concentration of remaining platelets in plasma prior to either downstream analysis or to freeze thawing. And we also recommend to remove platelets, possibly by this double centrifugation with filtration step. So another important finding from this paper was that even though the concentration of EVs is 3 orders of magnitude higher than the concentration of residual platelets in plasma, we found that due to their size, the remaining platelets actually have a similar volume fraction to our vesicles, which could mean that they, for example, carry the same amount of cargo. And this is where we laid the basis for the second paper, actually. So what is then the effect of those remaining platelets on downstream analysis? The goal of the second paper was to investigate whether those platelets affect the EV microRNA analysis. The paper has been accepted in the Journal of Extracellular Vesicles, and I hope in the coming weeks it will be online, so you can also view it there. And the first steps that we took for this paper was to validate the findings of the first paper, actually. So again, we saw that after double centrifugation, there were still platelets present. We also confirmed that indeed those platelets co-migrate with our vesicles during size exclusion chromatography. From my head, I think it was 20% that actually ended up in our EV-enriched sac fractions. And we found that filtration removes over 97% of those remaining platelets without affecting the EV concentration. And filtration can be performed on the plasma or on the EV-enriched sac fractions. Both work. The only thing, as I mentioned before, is that then the size exclusion chromatography should be performed directly and the plasma should not be frozen first. So then, of course, the main question, so what is the effect then on the detection of microRNAs? So for that, we have a collaboration with the other hospital in Amsterdam. And there they looked at 3 different microRNA targets. So 2 of them have been reported to be associated to EVs, LET7A5P and MIR21. And MIR223 is actually a microRNA that was reported to be associated to platelets. So this graph, I will get you through it. On the y-axis is the cycle threshold, and it's put in reverse here because basically a lower cycle threshold means a higher quantity of microRNA. So that's why I put it in the reverse order. And what you can see here is that in our unfiltered compared to our filtered samples, the filtered samples have a higher cycle threshold, meaning that they contain a lower quantity of microRNA. And we see the same pattern for all 3 microRNAs. Basically an increase of 1 to 2 cycle thresholds means a fold change in microRNA of 2 to 4. And that's why from this, we actually concluded that 50 to 75% of the microRNAs that we detect are actually originated from the particles that we have removed during filtration. So, for example, our platelets, but also other larger particles that we can remove. And then we decided to see, okay, is there a correlation between the concentration of platelets and the concentration of vesicles compared to the amount of microRNAs that we have present? So on the left side, you can see the concentration of platelets versus the cycle threshold. And on the right side is the concentration of extracellular vesicles with the cycle threshold on the y-axis again. The open symbols are actually filtered samples and the closed ones are unfiltered samples. And what we found here is that the quantity of the microRNAs is increased with an increased concentration of platelets. So you can see that here. Whereas for our EVs, or the EVs that we can measure with the flow cytometer that we are using, we did not observe the same trend. So in conclusion, for this paper, again, we found that double centrifugation does not remove all the platelets that are in our samples. These remaining platelets result in a 2 to 4-fold overestimation of EV microRNA, or microRNAs. One of them was not really EV associated. And we also saw that filtration can remove over 97% of those remaining platelets, again, without affecting the EV concentration. So what we believe from these two papers are the main important points is to really improve the reproducibility of extracellular vesicle research. We should definitely measure and report the concentration of remaining platelets in plasma before downstream analysis or before storage in biobanks. And second of all, those platelets can be removed using filtration. There are some discussion points that I would like to also highlight here. So first of all, this detection limit of the hematology analyzers. As I showed you in one of the first graphs, most of the samples that we will measure, plasma samples, would be considered to be platelet-free when we use these hematology analyzers. So when we measure and report them, we always use flow cytometry. Second of all, we only looked at 3 microRNA targets, and this effect can be different for each microRNA, of course. So that should definitely be investigated for specific studies. But also, besides microRNAs, this could have effects in other downstream analysis. For example, in proteomic studies, which is something that should also be investigated further on. What we would like to sort of stress is that the samples that have already been stored in biobanks for which we do not know the platelet concentration, it's really important to consider the utility of those samples in specific studies, depending on what you want to analyze, of course. As I already mentioned, our EV flow cytometer has a detection range of, and the analysis of 150 to 1000 nanometers. So the correlation that I showed you before between platelets and the quantity of microRNA and vesicles and the amount of RNA, we do not know what happens below this limit. So the effect of smaller vesicles and the concentration of microRNAs. And yeah, this is still work in progress, so it's still ongoing. One point that we are working on is the dead volume that these filters have. So to use these filters, we really need at least 1 ml of plasma or EV-enriched sac fractions. And second of all, this is really something from, I think, 2 weeks ago. We found that the plasma can have a maximum concentration of platelets prior to this filtration step. So if we would only centrifugate once, for example, and then use this filtration system, we saw that we are actually generating platelet-derived vesicles. So this is something really interesting to see the exact cutoff, like what can be the concentration of platelets in your sample prior to filtration. And this is something that we are working on, and we submitted an abstract also to the next ISAF conference. So I hope by then we can inform you on what happened there. And with that, I would like to finish and acknowledge all the people that worked on this study, all the co-authors, of course, Britta, but also the people in the lab and the whole team for helping out in these studies. So with that, I will hopefully open a nice discussion.
Wonderful. Well, thank you very much, Jill. Thanks to the whole team for this interesting work. So we do have some questions that have come in the chat box already. The first is from Nadim Tawil.
Hello, Ken.
Hi, thank you.
So I just wanted to follow up on this, and I know lots has been discussed that. So at the moment, if precious plasma is to be collected for multiple studies, including EV, circulating cDNA, there has been lots of reports of different speeds published, different times, different breaks with no breaks. Even there was one article that reported on one speed, if I remember correctly, was one spin instead of two, as a good replacement for the two-step approach. And what would be your approach of choice and your safest option if you know that at the moment you have to collect this plasma and there is clinical considerations to be taken? So in clinical settings, usually there is some resistance to changes of protocols. What would you go?
Yeah, fully agree. In our case, the samples that we collect are usually for extracellular vesicle research. So we really stress to make the sample as perfect for that as possible. However, of course, if a sample would also be used for other purposes, it really depends on the type of analysis, I would say, that is being performed downstream. For EV research, I would say to remove those platelets by filtration. As I said, you need at least 1 ml of plasma, which is quite a lot for clinical studies. So that is really something that we need to see if we can improve that. But for us, we want the sample to be as clean as possible at the moment. So yeah, I don't know if Britta or Rienk would like to add something to that.
Yeah, I can add something to that. So I would also, like in either case, go for standardized reporting. So whatever you're choosing as the anticoagulant of choice or the protocol that you're following, just report as many details as possible and be as transparent as possible. I think it is hard for us, as Jill already said, to give advice on use this and that tube or use that centrifugation protocol, because we don't know exactly what the downstream analysis is. But in either case, try to try to report as many details as you can, I think.
Thank you.
Thank you. Let's go next to Ellen Millman.
Hello, and really interesting talk. Thank you. One variable that you did not discuss is the choice of anticoagulant, whether it's K2 EDTA or citrate or heparin. Have you done any investigation of those? And the follow-on question is actually going to be, have you looked at the effect of the temperature of the centrifugations? For our research, for entirely different reasons, all of our samples have to be collected, put on ice immediately, and spun cold, 4 degrees. Have you seen any effects of that, or have you even looked at it?
I think you want to take it, or I can take it.
Well, I can say one thing about putting blood on ice. It is one of the most efficient ways to activate blood platelets. So if I would review a paper and I would see it, I would ask immediately to repeat the same experiment, doing it at 20 degrees centigrade.
Well, there for other reasons, other things that we're analyzing, we have to do it that way because it prevents activation of other things.
Yeah. Well, it is what it is. But for platelets, it is far from ideal.
Yeah, also commenting on that, so Jill and I ourselves did not look into differences between anticoagulants, but one of our colleagues, Naomi, did, and she has a paper out there on how EDTA versus citrate affect platelet activation and how the concentration differs. For example, and I know that Anne Hendricks' group is also working on a paper comparing different anticoagulants, but we ourselves didn't look into that, but we followed the recommendations at the times for platelet-derived EVs.
And you used EDTA, correct?
Yes.
Okay, thank you.
So I just have a follow-up on that. So if platelet activation is happening at 4 degrees compared with 20 degrees, what about 37 compared with 20? Why don't we keep the blood at body temperature?
That's a good question. Edwin would like that, because we have the idea that perhaps it's much more efficient at that temperature to separate platelets from vesicles. So that's a really good remark. We never did a follow-up study because we never had the centrifuge at 37 degrees in the lab. but who knows? We should try it. And just one more remark about the previous question. I'm not sure if we collect blood in EDTA, but you then would activate the platelets really at 4 degrees centigrade or at ice, melting ice temperature. Because EDTA is a real strong calcium chelator, and you inhibit both coagulation and you prevent full platelet activation. So perhaps that may save you there.
Artem Jivalozhny, you have a question about another kind of contaminant, co-isolate.
Well, thank you. Very interesting presentation. I was wondering about lipoproteins. As I understand, via filtration, you remove big contaminants, which is platelets. But what about small contaminants like lipoproteins? Because as I know, the amount of lipoproteins is terrific. And like, how you remove, or did you remove them?
Yeah, definitely. The filtration step is not going to remove any lipoproteins. Similar to not removing any vesicles. So in this case, this is really to sort of prepare your sample for either freezing or for downstream EV isolation. So for example, in the slide that I showed you, we first double centrifugate the blood until it's plasma, we filtrate the plasma, and then we perform SEC with the filtered plasma to isolate our EVs. In that way, we can reduce the concentration of lipoproteins, but yeah, there's still a lot of lipoproteins present even after SEC. So the filtration will definitely not remove any lipoproteins.
Yeah, that's a completely different universe from the large to the small, and lots of diversity in the lipoprotein world as well. Lipoprotein world. Hopefully, Wyatt Freeland is not listening to me mispronouncing his favorite word. Okay, so let's go to Louis Vu. Is it Louis or Louis?
Hi, it's Louis. Yeah.
Okay, Louis, go ahead.
Thanks for the nice talk. I just have a question, because actually for my research, I'm more interested in preserving the platelets from fresh samples. So, and I have done several rounds of platelets isolation, and I think I don't know whether you guys would want to dilute the plasma before the platelet removal, because I think that helps a lot, as the plasma is quite viscous, and that kind of like suppress the efficiency of the centrifugation. Also, I feel like the protocols that you show, they use very high speed, only one that use around like 900 G. And I was wondering if that is also a factor that could, activate the platelets, and because higher speed would create like shear force, and that would kind of like automatically activate those platelets.
Yeah, so for the platelet activation, as Rienk mentioned, in the EDTA tubes, we do not see platelet activation either before or after the filtration step when we use this protocol that I showed you on the slide, so the 2500 Gs. With diluting the sample, do you mean the whole blood or the plasma?
So after you spin the whole blood, right? When you get the plasma, I would dilute it, because that's based on the protocol I have for platelets isolation. But also, just a follow-up question: what are the markers that you use to detect platelet activation here?
Yeah, so in this case, we use a combination of CD61 and p-selectin, so CD62p, for platelet activation as platelet activation marker. For the platelets themselves, it's CD61 and CD41. And yeah, diluting the plasma could be an option, especially in the samples that have too high platelet concentration to say. The only thing I can think of downstream is that maybe in case of, for example, microRNAs, of course, if we dilute the sample, then maybe we also get into problems there, but we could concentrate it again. It's an option that we could try in case of samples that have too high platelet concentrations.
And just one final question is that, so do you include a positive control in your platelet? Because, if you activate your platelet with, I forgot the name of the reagent, but when I do add that into like the platelet breaths, they do activate a lot, and I could see like a clear shift in the signal of like CD62P and CD63 in my panel. So
also in EDTA collected tubes?
Yeah.
Ah, okay. Well, it happens that 2 or 3 weeks ago we tried this, but I barely saw any platelet activation in plasma that was originally collected in EDTA whole blood.
Well, okay.
But it's yeah, something to look into. Thanks.
Thanks for the questions. Now, Derek Allen Haas has asked, have you tested the use of PGE1 to prevent activation and prevent the release of these platelet particles? And Rienk, you've already answered that no. But Derek, do you want to expand on that? Can you give us the background for this question?
Sure. So when we had been working on vesicles from patient samples, we had worked in conjunction with an actual platelet lab. And so one of their protocols was to initially dilute down the blood in citrate solution 1:1, and then we spin them only at a very low speed, similar to what Lou said, maybe 100 G to separate out and isolate the platelet-rich plasma. From there, we added about 1 nanomolar of PGE1 to prevent the platelets from aggregating and activating, and then proceeded to pellet the platelets before taking the plasma to kind of spin it down further for the isolation of vesicles. And that seemed to get us a much lower number of vesicles compared to if we didn't treat it that way. Because again, like it seemed like if we, what Lou said, if you spin them at high speed, it seemed to cause release of the platelet microparticles.
Nice. Yeah, it's well, in terms of platelet activation, I'm not very experienced, so I think Rienk has definitely more experience in that.
Yeah, well, if you add it, you will increase cyclic AMP in the platelets, and you will make platelet activation more difficult. And I think it's a good question because it shows that yeah, perhaps we do need many more studies on optimizing these protocols. I mean, if I look at what Jill did, we see that platelet concentrations that are about 100-fold below the lowest concentration of platelets that we can measure with a routine analyzer. And we still see that it explains 50 to 75% of the microRNA that we detect and that we think that's associated with the vesicles. I think many labs will have much more platelets than we have around when they're measuring microRNA or whatever. So I think there's a lot of optimization and standardization that we can do to improve downstream measurements. And of course, adding inhibitors may also help, or choosing different anticoagulants or centrifugation at different temperatures. I think there are endless lists of things we can vary there. Sure.
Thank you.
Next, we have a practical question from Clotilde Théry. Clotilde.
Yeah, hi, thanks. So yeah, I wanted to come back on your recommendation to report the number of platelets before freezing the plasma. And actually, it's particular because I tried to implement that in our hospital, who's collecting plasma more for DNA, ctDNA analysis. And when I told them, well, can you analyze platelets there before freezing? They told us, no, it's not in the protocol. So I was just wondering, I mean, if quantifying somehow platelet markers afterwards, when you isolate the EVs and when you analyze the EVs, would not be a good option, or maybe, yeah, as good, or maybe even better, I don't know. After
freezing, yeah, I think the only issue then would be that you don't know what the sample looked like prior to freezing. So you generate platelet-derived vesicles, but then maybe that's different in each sample depending on the concentration of platelets that was in there. But then you would like know how much platelet-derived vesicles you would have from, I mean, in the EVs that you analyze, so that it's a more direct measurement of the potential contamination, isn't it?
Yeah.
Nah, but you cannot, like, so if you don't have the initial platelet counts or concentration, you cannot, like, say one platelet created so many platelet-derived vesicles, right? So I think you would need to have both, but if the only way that you can implement it in your lab is to measure it after, but you're aware of this and like comment or like put a comment or whatever and like you're reporting.
Yeah.
I think that's fine as well. Like, because we also know, as Jill said, like for some of the studies that are already ongoing, this is just not a possibility to measure either before or after anymore, or even using a hematology analyzer, which may look like your sample's actually platelet free, which is not if you measure it on a flow cytometer. So I think it highly depends on what's really feasible in the setup of the study itself.
Yeah.
Yeah.
Thank you.
Yeah, Clotilde, maybe I can add something so now and then we get samples from studies from other research groups. They ask us to look at vesicles in blood plasma that they collect, and we always ask them to collect blood from a few healthy individuals in their lab, prepare the plasma, freeze the plasma, send it to us. We analyze the vesicles on the flow cytometer, and we also look immediately directly at the platelets. We can easily see them. And then we have a very good, idea about whether they are really capable of removing most platelets or not. You immediately see a big platelet cloud or not. So it helps, at least to me, it shows that the procedure of blood collection, handling, and platelet removal works. So it's more about quantifying confounders. I mean, platelets are really difficult to get rid of. Density gradient doesn't work, SEC doesn't work, etc.
Thanks.
Thank you, Clotilde. Berta Puig.
Yes, hi. Can you hear me?
Yes, we can.
Hi. So very nice talk and very useful, really. I just want to follow up in this thing about not putting the blood in at 4 degrees or on ice. So if we cannot do that and our hospital is a little bit far away, which time will you say between collection and centrifugation can pass, more or less? Like if I report this in a paper or something. So which time would you say it's okay? Or
Jill, shall I answer this question or?
Yeah, go for it.
Well, it is. I think it's. First of all, I think the most important thing is that all samples that you collect are treated in the same way.
Okay.
So I think that's really essential. So if one sample takes 10 hours and the other 25, then I think you will have incomparable results. The other thing is that if you use an anticoagulant like EDTA and it is compatible with your downstream analysis, routine laboratories all over the world use EDTA blood to measure the concentration of platelets. And the concentration of platelets that you measure in EDTA blood, at least our routine lab claims that it's stable for at least 1 week. That means that the platelets are not really activated, and I think they are unable to release many vesicles or not vesicles at all. So I think it really depends on the tube you're using, the anticoagulant you use, the way you standardize your protocols, etc. And of course, in real life, not everything is possible. The standardization, treating all samples the same, I think that's where it all starts. And choosing the right anticoagulant, etc.
Yeah, okay, good.
Yeah.
So, but if we are talking about half an hour, 1 hour, and we more or less do the same procedure.
No, that's fine. But again,
fine,
also there, if you can collect the blood samples, if you have the opportunity from fasting individuals, let's say in the morning, whatever, everything you can standardize will help you to get more reliable results, I think, in the end. But of course, it's not always possible in clinical practice.
Okay, good. And the important thing is report anyway, no? To report the protocol and how we have done it. Mm-hmm.
Yeah.
Okay, good. Thank you very much.
Yes.
Thank you. We have a question next from Jahangir. So Jahangir, you have asked about a sandwich immunoassay. So specifically, what variables are you concerned about here?
Yeah. So typically for all the immunoassays that we have developed, we have measured the protein concentrations in serum or EDTA plasma, and protein concentrations for majority of analytes turn out to be almost the same concentration. So we haven't seen a huge impact of platelets that are remaining in plasma. I clearly see that platelets have impact on EV measurement, but has anybody come across the challenge that is faced with EVs versus protein measurement using immunoassays?
So if I understand correctly, you're asking if, so you say platelets do not affect the protein concentration that you're measuring?
Right. In majority of the cases where, for example, we use serum or paired EDTA plasma samples from the same individual, and with the immunoassay, we can measure the same concentration of the analytes in majority of the cases. So we at least did not see the impact of remaining platelet in plasma. So I clearly see that the EVs are impacted by the presence of platelets, but I haven't seen that for immunoassays for protein measurement. Has anybody come across a situation where the protein concentration is different depending upon the platelet concentrations in plasma?
Yeah, no, we haven't looked at it. It's something that we are considering, because of course after microRNA we would look at other downstream analysis as well. The only thing I could think of in a case, because what you mentioned is that you have serum and plasma from the same patients, but right. Then in case of serum, maybe the proteins are actually released, right? So
yeah, the proteins are released, whether it's, I mean, they are basically released into blood, right? Yeah. So whether you use serum or EDTA plasma, or in some cases even the citrated plasma, we see almost identical concentration among different blood samples.
Yeah, that's interesting. No, we haven't looked at downstream protein analysis. But yeah, it's something that would be worthwhile, I think, to also investigate further on what would be the effect of remaining platelets there, definitely.
Now we've come to the end, just one more question left, and this is a question from Pia Siljander. Pia, please go ahead.
Hi, it was just more like a query about the ACD compared to what you see in comparison to EDTA and citrate. So citrate is not good compared to EDTA, but I'm not a fan of EDTA either. So I would go for ACD, but you have a strong preference for EDTA. So I wonder what you see with ACD. And have you actually looked at, referring to the previous interesting question, in time, what Rienk was saying, that there is no EVs coming in EDTA samples in time, that have you actually looked at it? So I think it's really like relevant.
Yeah, it's a good question, Pia. So I think using more and different anticoagulants, trying to optimize the blood collection and the plasma preparation protocols, I think it fits in there. I think ACD is milder than EDTA. We have compared the platelet activation in citrates and EDTA over time, and you clearly see that platelets become activated when you have, let's say, if you have a tube of whole blood that you put on the table that's collected on citrate, you see platelet activation and platelet disappearance, and you don't see that in EDTA. But of course, EDTA is a very strong anticoagulant. So yeah, perhaps ACD is better. We didn't try it. I just don't know. But again, I think it fits into what Ken said: centrifugation at 37 degrees, adding PGE1 perhaps to block platelet activation. Different anticoagulants. It may all be useful to test. The only thing is there's a lot to test, and this was just a sort of, I think, preliminary study. but at least it shows that it does make quite a difference, how you treat your samples and the purity and the quality of the plasma that you prepare. So yeah, I'm sure it can be optimized, and perhaps ACD is a better choice. I don't know.
Well, ACD is the acidicity pacifies platelets, and also the dextrose makes them happy, so they are least likely to do something. Of course, if you can really handle, then I think the PG1 probably is a good idea, even the dilution, whatnot. But I was wondering, because you recommended, or sort of I understood in your comment to Britta, that you could even analyze EVs from a week-old EDTA sample. So I don't think that's...
No,
have you actually
not
tested that?
No. But the only thing is that if you would have a tube of citrate blood that you put on the table... Not
citrate, ACD.
Yeah. But yeah, ACD. But yeah, okay, I don't know. We should test it. Simple as that.
Yeah.
Yeah.
And I see a comment here from Naomi about kind of backing this up with the ACD blocking activation. However, there is a practical consideration that EDTA is more commonly used in the clinical samples there. So Naomi, did you want to expand on that? You don't have to. I mean, it's very clear from your comment, but thanks for sharing your observations too. And we have Alain. Alain, I have to apologize. I just saw your sign up at the very last minute here. So thank you for joining. And I'm glad that you could join. But you've had some different experience here. Could you let us know about that?
Yeah. Well, hi everyone. I was quite puzzled by the results about the number of platelets remaining after the 2 cycles of centrifugation. Well, we do that routinely, and we don't get that many, far from that many platelets. We have an average value of 30 platelets per microliter, within very rare occasions 100 platelets per microliter. So it is, very, very far from at least the study A in this paper. And I was wondering whether there could be probably a problem with the centrifuge. We getting that many platelets. That's not, in a sense, it's not possible.
Yeah, you caught us there, I'd say. So that's something we commented on in the paper as well. The centrifuge was in fact broken, but we would have, so on paper, we used the exact same protocol, and without comparing it to the other centrifugations that we did in the other studies, we would have never found out that the centrifuge was actually broken. So this also shows that even if you report this and say on paper, this is how we use it, that's not always, like, there are steps in between that can just go wrong. But yeah, very well seen, I'd say.
Okay, good. Yeah.
Oh, so there's an explanation. All right. Very good. All right. Well, we have come then to the end of our session today. Thank you to everybody for the insightful comments and questions, and special thanks to Britta, Rienk, and then Jillian first and foremost for presenting the work today. We're very happy to have learned from you about platelets, and we look forward to your continuing work in this area that's, I think, is very productive and very helpful for the field. So thank you all. We look forward to seeing you again at a future EV Club, and I hope you all have a great rest of the week. Take care now.
Thanks.
Thanks, Ken. Thanks all. Bye.